by our friend, Dr. Fanos Alexiou. This is in the context of the challenge, the quantum formalism challenge, hackathon challenge that we are running. There are actually two challenges. One of them is on, federated implementing federated learning in a quantum setup, taking into account existing NISQ devices. And the other one is on encoding, classical to quantum data encoding. In particular, one of the things I suggested, or at least I'm curious to know, is imagine the classical data set sits on a rich geometric structure, like, say, for example, a manifold structure, let's say a symplectic manifold, for instance. And then the question is, can you find something, a new way, can you hack your way through something new to map the data sets into quantum states on a Hilbert space? So that's essentially, in a nutshell, the challenge for the encoding. So another thing we decided to do is to kind of have some sort of a use case in mind, rather than just a generic type of a challenge. Obviously, federated learning and also the encoding can be applied to many use cases, but we wanted some use cases, a specific use case. In this case, we picked genomics because it's a very interesting field, as you will learn from our presenter today. So yeah, so before that, let me introduce our speaker. So thank you so much for taking your time, and also for putting yourself available to, on Discord. Once the hackathon challenge starts, you will be on Discord, helping out the teams with any questions and answers about, genomics related stuff. And hopefully you'll find it interesting to see also, some of the stuff that people will come up with on the thing during the challenge. So yeah, so thank you, Fanos. So Fanos Alexiou, Dr. Fanos Alexiou is currently the ERA Chair in Bioinformatics for Genomics at the University of Malta. He studied genetics as an undergrad degree at University of Aberdeen in the UK, in Scotland. But he quickly fell in love with the emerging discipline of bioinformatics. He completed his master's at the University of Amsterdam and a PhD in Greece on bioinformatics. After his PhD, he moved to the University of Pennsylvania School of Medicine. This is why we were talking about the Eagles, the Super Bowl, because he lived there during his postdoc. For his postdoc research, he was focused on bioinformatic analysis of next generation sequencing data. And in 2018, he returned to Europe as an independent researcher at the Central European Institute of Technology, where he led a team focusing on development of machine learning algorithms for bioinformatics. And since January 2013, he moved on to a new challenge, which is to bring structural changes at the University of Malta, establishing and nurturing a strong bioinformatics community. This effort is supported by an ERA chair grant called Biogen MT. And yeah, it's great to have Fanos with us. Fanos has published over 20 scientific research articles as well. And collectively, they're being cited over 5,000 times, So he also has been the principal investigator of several national and international research projects, such as deep learning for genomics and transcriptonic pattern identification, and so on. So it's great to have Fanos with us today, and I'm pretty sure he will inspire you, for those on the challenge, to look at what he has to share, and hopefully inspire you to come up with something novel. So without further ado, Fanos, thank you so much again, and the floor is yours.
Okay, nice to be here. I'm actually, I don't know much about quantum computing at all, but I'm really, really interested to see what people will come up with. And I'm really, I will definitely be very, very hands-on and available if people have questions. So my assumption here is that people don't know much or anything about biology, and I assume that people might have some knowledge about machine learning. So I made a presentation trying to inspire a little bit, like the general ideas of the field, and then give a base of biology or genomics. And finally, I'm going to talk about the data set that we will be sharing with you for the challenge. So um, at least I hope everybody has heard of DNA, the blueprint of life is a biological molecule that is found in every living organism on Earth, and it encodes all the information needed for living cells to produce every single thing that they need to operate, uh, in life, right? What we will be interested in today is trying to identify patterns on DNA that produce specific functions. So we will get into it a little bit later. So the story, I will give you a little bit of a historical background on on this science. The story starts about 200 years ago um, with a monk, I mean, he was more of a teacher than a monk, but Gregor Mendel, who started noticing interesting patterns in how peas, the plant, the color of their flowers and of the shape of their fruit, or of the beans, was differentiating based on how the plants were being crossed with each other. And back then, of course, nobody knew anything about DNA, about anything like that. And the world was not exactly ready to absorb this information. Um, so the idea that heredity is somehow carried on through something that it was not easy to define back then was forgotten for about 50 years. And then in the early 1900s, new disciplines started emerging, combining research from chemistry, from physics, and from biology that were already established disciplines, and start to creating the sciences of genetics, the science that studies DNA and heredity, biochemistry, so chemistry and biology, and biophysics. So the theme that you will see again and again mentioned in my presentation today is combining already established fields to create something new, and this something new can take on the challenges that are created from our deeper understanding of the respective fields. So interdisciplinary research and at that. So we will jump a little bit forward to the 50s, and now we're seeing the start of parallel development in genetics, where famously Watson and Crick identify the structure of DNA and identify that DNA itself is, like how the information is encoded in DNA, for which, of course, they received the Nobel Prize. At the same time, in the US, the first machine learning computer programs have started being developed. And the idea is that these are programs that can learn from previous information, and they can learn how to improve themselves into to do some specific task. So the most important thing, if you don't remember anything about biology and you don't need to know much about it, the most important thing to remember is that there are three layers there are three layers of how information flows in a cell. There is the DNA level, the RNA level, and the protein level. The DNA is the blueprint. The DNA, every cell in your body has the same DNA sequence, which is about 6 billion letters long, and it's the same in every single cell of your body, which means that, for example, a skin cell or a brain cell that are doing completely different jobs have exactly the same starting information. Okay, this information needs to be activated and to be passed on to producing functional tools. The functional tools are the proteins. So proteins are pretty much everything in your body is built, I mean, roughly speaking, by proteins, including being the building blocks, like the bricks, and the machines that put them together. The intermediate step between these two things is the RNA, and the RNA is very important because it's both the short, it's the way that information that exists in DNA is encoded so that it can produce proteins, but it's also in itself functional. So there's RNA that can that can act as a molecular machine and can do things in the cell. Roughly speaking, and kind of what we will focus on today, is parts of the DNA that produce RNA that are called genes. A gene is a part of the DNA that produces one RNA molecule. And generally speaking, one RNA molecule produces one protein molecule, or can produce one protein molecule. Okay, so moving forward a little bit with our historical, with our history, in the 1950s, we are starting to have this very complicated artificial neural networks that were still analog at the time, and there's a big hope that using these very basic artificial neural networks, eventually this type of networks will be able to learn enough to complicate things like understand language, translate, make decisions, and so on. And in the 60s, it's shown that using this type of neural network, simple problems such as linear regression can be solved. Now, of course, going forward a little bit, this hope doesn't pan out, and millions and millions of, of dollars are spent, especially in the US, to try and bring perceptrons or the simple neural networks to do more and more complicated things, and it becomes evident that this will not happen. So in the late 1960s, we have an AI winter, meaning that research funding dries out and the progress doesn't follow as fast as people hoped. But what happens in the 1960s, in the late 1960s, is that a new combination of sciences comes together, and a new science called bioinformatics is created. This is, let's say, a mix of biology that already existed and the emerging science of informatics. Of course, a small part of it is machine learning that we're more focused on today, but the other things, bioinformatics it's a very wide science today and has many applications. The beginning of bioinformatics in the 60s was looking at proteins and trying to predict protein structures. So proteins, as we said, are the more functional molecules, the machines of the cell, and the way they look, their structure, in many ways defines their function. So being able to predict how a protein folds um, is not perfectly equivalent, but is a close approximation of figuring out what it does. So that was that was one of the first, let's say, fields that people were interested in back in the 60s, is trying to predict protein structures. And the first book of protein structures contained the 65 protein sequences, which at the time was a huge number. They published even a book uh, for it, which kept being published every year with more and more structures. Of course, today we have hundreds of thousands of structures available. Um, so um, in the 70s, again, people kept working on protein structures. That was like a big focus, and they started kind of um, again figuring out their different types of substructures, and that the big concept was that in some way that we cannot predict right now, the sequence that comes from the DNA into the RNA and into the protein sequence, the sequence itself should contain all the information to define how the structure of the protein, the physical structure, should look like. And this became at the time and until recently the kind of holy grail of bioinformatics and machine learning, especially in the field of bioinformatics, trying to figure out um, if you're given a protein sequence, so you know the gene that encodes it, how will this thing look physically? Just a reminder. So in the 90s was created CASP, that's the critical assessment of protein structure prediction, which is basically a benchmark. And how this worked was that every 2 years, the people organizing this would experimentally, in a very difficult process and very expensive and difficult process, they would experimentally figure out the structure of a protein or a handful of proteins. They would tell beforehand. They would tell all the machine learning people what these proteins will be, and then everybody that want to take part into this would try to predict the structure, and then they would get validated based on the real experimental data. So this was kind of having this benchmark is what really propelled protein structure prediction as the one of the biggest and most let's say, when it came to machine learning, pretty much everybody or most people were working with protein structure. And in 2020, we reached a point where, with the new machine learning techniques, the problem was finally solved. So for 50 years, basically, the whole community was working on finding a solution to this problem, and with many dead ends and kind of getting to a point, but not further. And a couple of years ago, Google came up with AlphaFold 2, which essentially at this point has reached, is basically as good as the experimental techniques for most things. And of course, it's free compared to an experimental technique that could take months, and possibly thousands or tens of thousands of dollars to get a structure. You can now do it in a few minutes in your your computer. So this basically completely revolutionized the field, and now it's one of the few times, at least in my experience, although it's becoming more and more common, that we have seen, let's say, holy grail getting solved so completely that even the competition that has been happening on now needs to change and start giving different targets, different tasks to the competitors. I will not go into too much detail about how it works. I just want to show you. I don't know if you can see my. Yeah. I just want to show you just the progression over the past 15 years. Um, these are the winners of each year, and the 100 means that they would get perfectly accurate prediction based on the experimental results. And for many years, it was around 30 to 40, depending every year on the proteins. And then you see within 4 years, AlphaFold and then AlphaFold 2 just like went through the roof. Over 80, I believe, is considered as good as it gets when it comes to accuracy. Okay, so yeah, and then of course we have the big, everybody, everybody starts using this now. There's several branches of protein structure prediction with various applications that come from that. Okay, now this was kind of a short like story to show you that machine learning in biology has a long history, and we are really now just starting to get the benefits of the big developments in machine learning applied to these big biological problems. Again, we were talking about proteins, and now we will start going more towards what our focus in genomics, which is the DNA. So jump back to the 70s. In the 70s, for the first time in the late 70s. So remember, in the 60s, DNA was discovered that exists, and the structure of DNA was discovered. But in the late 70s, we actually started being able to read the sequence of DNA, and this brought the idea that eventually we could sequence the whole human genome or genomes of other species. Of course, in the 70s, and you see here the actual first paper on one of the first papers on sequencing, um, something like reading about 5,000 letters of DNA was considered a large amount of data. Imagine a human genome that is 6 billion bases, 6 billion letters, right? So that was uh, science fiction at this time. But in the 80s, so a few years later, um, um, we started seeing automated genome sequencing becoming a thing. So things could be, it didn't need to be done so much manually anymore. Getting machines to do the actual sequencing, and of course, then this starts to allow we have more computing power, so you can get more um, you can store larger amounts of data. You can do things like align sequence to each other and so on and so on. In the 80s, by the way, we have another AI winter after the expert systems idea kind of dies off in the late 80s. And in the 90s, finally, we get the idea that we should sequence the whole human genome, like we should go for it. And it's a 15-year project that is funded by many governments around the world and mostly the US. And the idea is to basically try to use the techniques that exist at the moment, but also develop new ways to accelerate this project and to sequence every letter of the billions of letters in from one human genome. Um, and at the same time, in the machine learning field, there are new techniques being developed, but still, why are governments interested in sequencing the human genome? So I will get to that. I will get to that. So why does it matter? Why do we care about how what a genome, what are the letters on a genome? As I said, the letters of the genome encode all the information that is found in a cell. Um, for example, there can be diseases, and there are many, where a single letter could change, and this change could lead to, for example, a genetic disease, or a few changes could lead to cancer. Knowing what the baseline is, so knowing what the genome should have there, allows allows us to, for example, to test for this type of disease, or even to identify what could go wrong and lead to this disease. This is just one use. Why the government would fund that is because this took an enormous amount of money, and it needed collaboration of labs across the world to over a decade to get it done. So nobody else could really do it. But we will look at that now. In the late 90s, as parts of the genome start getting published, um, people in the bioinformatics field start creating machine learning models to try and find which parts of the genome encode genes. So which parts of the genome eventually give functional proteins or RNAs. And this is not as simple as it sounds, because most of the genome we came to understand when we, this started happening, most of the genome actually doesn't encode genes. It's only a small fraction of the genome that encodes genes. So in many ways, the genes are kind of lost in this background. So we will go a little bit deeper on how a gene looks, but the general idea is that it's a piece of it's a sequence of letters. The length can be variable, very variable. It could be even, it could start from something like 100 letters and could go up to millions. And generally, these letters are organized in blocks. Some of them are called exons and some of them are called introns. We'll talk about later. What will be important for our task today is how this expression of this gene is regulated. And much of the regulation of these of genes happens at the promoter region, which is basically the control center of the gene. So if a gene should be expressed a lot or not is controlled in many ways from the promoter region. So we will be the task for today or for this week will be to see if we can identify promoter regions. Okay, so as I said, in the late 90s, bioinformatics starts trying to predict genes and try to predict, trying to crunch through genomic sequences as they're being produced. And finally, in 2001, a few years also, I think, ahead of schedule, we get the human genome sequenced, and actually we get that from two sides. So we get one, the big international human genome sequencing consortium, which is like the list of authors is here. Sorry, no, it's a different thing. And this is like this big international consortium that has the governments and research institutes and so on, that took about 10 years to produce the human genome. And then in the meantime, because new technologies were being produced, an independent organization started in parallel a bit later with private in a private initiative, and they ended up finishing at the same time. So they essentially published their their genomes at the same time in February 2001. One was published in Nature and one was published in Science, like the two big journals. So this really brought us with a new millennium, brought us to the genomics was revolutionized because now we had the genome and we sequencing genomic sequence started becoming much cheaper, and you can see that the human genome, of course, the human genome would cost about $100 million to sequence in 2001, and then during the 2000s the price started reducing this logarithmic scale, by the way, reducing, and then you see an inflection point around 2008 when this next generation sequencing becomes available, which I will not bore you with, but is this type of machine that can start. Of course, now we have even better ones that can start sequencing a whole human genome in like I think right now it's maybe a day or maybe less, and we have definitely broken the $1,000 price point by now, and it's if you sequence in bulk, it's even way less. So now we're at the point where instead what people are doing is going sequencing hundreds, thousands, of genomes to start doing comparative studies, to doing things like that, or sequencing other species, which is very important also. So you see here the increase in this is uh, excuse me, this is genome sequenced. Um, of course, it's in the thousands because we're talking about bacterial genomes that are very small. Um, but even you can see the orange line here is eukaryotes, which is like more advanced organism, and you can see like everything, all the lines are going up exponentially. Pretty much we have I don't know the exact number, but definitely hundreds or thousands of genomes sequenced every year, and for higher, let's say, organisms, definitely tens every year or hundreds. The problem is, and here's where this question comes in, you can sequence a genome pretty cheap right now for 10 years or more now. The problem is that annotating the genome, like sequencing is one thing, but then annotating it to say what part does what, this is what we're interested in the end, is not easy. It's expensive to do it. To do it experimentally, it's almost impossible. So we are building, I mean, everybody's building machine learning models that can take the information we have from well annotated species like human and apply it, for example, in other species. Um, the other thing is, I'm talking about only DNA sequencing, but now we are having this type of method, experimental method for sequencing, has now been is branching out into tens to hundreds of different methods, and each one can measure different types of biological data, um, giving slightly different information. So this is a very exciting time. The previous decade, where sequencing technology got diversified. Um, a notable example is in 2012, a huge project called ENCODE, where again labs from all over the world came together, and they took the human genome, and they did as much annotation as was possible at the time. And I think, I don't remember the exact numbers, but I think it cost close to $1 billion overall, of course, over years. So they used many different technologies, and they annotated as much of the genome as possible. And here you can see again a gene here, and then this yellow area represents the promoter of the gene, which is what we will be working. So meaning there's lots and lots and lots and lots of genomic data. The big question is, it's impossible to parse by humans. We need to build machine learning models to start understanding, building models, and expanding what we know. Another question is how much of the DNA is junk, because whatever we don't understand, we call junk DNA. You probably heard the expression. But it seems like we're starting to see that more and more parts of of our genomes actually do things, but the things they do might not be as obvious uh, based on the understanding that we have now. And much of that is regulation. So maybe a region of the genome could be in some way acting to regulate another part in a kind of network, another part of the genome, and in that way to affect, for example, how much a gene will be expressed or not. So we're figuring out that more and more and more layers of regulation in the genome. Now, we're talking about promoters, but what we're learning on well-studied things like promoters is also being applied to newer things that we don't completely understand, like different types of regulatory elements, for example. In the past 10 years, we've had a new revolution in machine learning, the deep learning revolution. You probably have heard about this. I won't go too much into it. The idea being that you have um, neural networks with many more layers, and also with different type of layers that can better capture patterns in data, and they can importantly capture patterns from mostly raw data without the need for um, for feature for making too elaborate features. That's something that we had to do um, before this kind of deeper neural networks became available. Of course, these are used. There are many applications in the world. We already talked about protein folding here. In biology, it's also used for image uh, sorry, for medical imaging, and of course it's used to do anything like natural language processing, um, producing art, or nowadays text, code, and so on. So these technologies are going everywhere today, and of course, bioinformatics is no exception. So we're getting a lot of new techniques that are very helpful and are really revolutionizing the field in the past 10 years. So the question we're going to talk about today, in a way, it's I like to think about it as finding a hairpin in a junkyard, not a haystack, because a haystack is relatively easy. In a junkyard, there's many more things that look like a hairpin, and you can, it's all metal parts that could look exactly like the thing you're looking at, but they are really not what you want. So in a genome, let's say a human genome of 6 billion letters, imagine, excuse me, imagine that there might be 100,000 genes. Let's say roughly, if each gene has a promoter, um, this means that there's like 100,000 promoters out of 6 billion letters. This really it's a very small part of the genome. So you really have to take into account that you are working with a very heavy class imbalance. So there's many more negative uh, class samples than real positives. Which means that generally, why I'm saying that is that generally in this type of applications, we try to care more about precision than about recall sensitivity. Um, It's fine if you can get every single one, but you really don't want to get false positives. Also, in many ways, you don't know how things look. So it's really hard to make features, which is great. That's why deep learning applications are great, because you don't need to predefine the features. So that's a kind of small graphic like about deep learning. Um, we can use basically more layers of a genome or more layers of a network instead of an expert telling us exactly what the feature should be. Okay, so coming to the actual data set, we have not published this yet, but there's a preprint out. So you can go, if you want, you can go and read that. There's not that much information in it, but the basic information is like what is in the data set and our application of a very simple convolutional neural network that we use just as a baseline so that people have something to benchmark against. The important thing is that we have made it as easy as possible for people to be able to download this data and build their own model. So, I mean, we were more thinking that people will be using different deep learning architectures on that. But now, this came up, and I will be very interested to see what you will come up with. So what is the data set consists of 36,000 sequences, DNA sequences. Each sequence is 251 letters long, and the alphabet of DNA consists of 4 letters. So it's 251 letters long. Each position could have one of the 4 possible letters. And this was originally published in 2017, but we have uh, taken the data from this publication here, and we have kind of repackaged it among other data sets to make a benchmark suite. So I picked this one because this is the most straightforward to explain what's happening. But if you go to this link here, you can also download other benchmarks that we have, and we have again done the convolutional neural network on each one of them, so there's a baseline. And I don't know how much. Okay, what is the nature of the class? So I think it's my next slide actually. I have there are 2 classes in this data set, and the first class is promoter sequences, and the other class is non-promoter sequences. I will tell you the exact details in later slides, a couple of slides later, and I will explain exactly what the promoters are. So, as I said before, we have here the little schematic. We go from DNA to RNA to protein. The protein is the actual functional thing that we want. This whole process can be controlled at various stages. And basically, what we want for example, a cell might want a lot of gene 1 and not a lot of gene 2, as an example. So it will want a lot of protein 1 and not much of protein 2. So what will happen is that the promoter of gene 1 will be highly activated, and the promoter of gene 2 will be silenced. And this means this will lead into high levels of transcription of the product of gene 1, and then eventually this will lead excuse me, to accumulation of protein 1, and the opposite can happen for protein 2. So the promoter, and and the enhancer, which we will not talk about today, but it's a different, it's a similar but different thing. These are functional locations on the genome. They're made out of nucleotides that control, modulate the level of uh, transcription of genes, like how much of this gene is produced downstream and goes towards protein. You can think of these as control panel, like analog control panels. So a promoter looks traditionally like this. It's a sequence of DNA. It has a length of approximately 250 letters, and on that promoter there are regions that are not always in the same in the same position, that are called transcription factor binding sites. These transcription factor binding sites are docking stations for proteins that are called transcription factors. There are thousands of these proteins now. When a transcription factor binds to the binding site, to its binding site, it does so by recognizing a pattern in the sequence. This pattern is short, could be from as short as 4 nucleotides. Usually they're around like 6 to 8 nucleotides, this pattern, so it could be up to 12. And is degenerate, which means that usually slightly similar sequences could still work, maybe work a little better or a little worse. So they're very, very hard to identify accurately. And what happens is now this transcription factors binding to the promoter, if they bind or don't bind, this can modulate, like it's basically turning the knobs of the controller, and this can affect how much of this gene will be produced or not. Many promoters also have a thing called the Tata box. The Tata box is a very well conserved, like part of the promoter that is found uh, 30 letters ahead, before the upstream of the start of the actual start of the gene, and it has a very characteristic sequence that is TA. So one sec, I will get the answer. So what we will be dealing today, we will not be looking at Tata promoters that are much easier to find because of the TA sequence. We will be looking at human non-Tata promoters. Okay, so it's basically promoters, and there are thousands of them that do not have this Tata box that is easy to find. These transcription factor binding sites can be in many different orientations. There are thousands of them, so you can really go exhaustively and look at that. So the dataset has thousands of sequences. Each one has 251 letters. And there's a positive class that is real human non-Tata promoters, and the negative class that is the same size, but random parts of the genome that are not promoters. So it's a pretty straightforward classification task, basically, what we're trying to do. We have also already split the data in training and testing. You will see it in the when you go in the GitHub repository. So if you want to, I mean, you feel free to split as you want if you want to try it. But if you want to really test, let's say, against our benchmark, this is the split that we use, so you can really reproduce. Um, and yeah, and we basically, as I said, we produced the convolutional neural network as a baseline. We didn't really go crazy like optimizing everything, so it's just there for people to have something, to have an idea about how accurate someone can be with pretty easy approach. Um, so and this is the end of my presentation. The full paper is here, and this is the subfolder within the. So this, if you follow this, it will take you to the GitHub repository, and this is the subfolder where there is a how's it called, the Jupyter notebook of Python that is an example of how you download the data, and then we have also the training of the convolutional neural network, and there you can also see how long it takes to run. Each epoch takes about 7 seconds, so it's pretty fast to train. Yeah, and there you can find, in this repository you can find all the data, and be able to download it. I'm not sure if what programming language you use or how you work with data, but from there you can export it as a text file essentially. There's also a question that maybe okay. So there's one question first that says, "Is any of these features controllable?" I'm not exactly sure what that means. The other question says, did you need a significantly large in terms of width and length CNN model? Let me remember what we used. I think we used, it's in the paper, but I think we used 3 layers, 3 convolutional layers, and then a couple of fully connected layers. So it was not particularly deep in that sense, because there are models with uh, hundreds of layers. No, it was a pretty shallow convolutional neural network. But if you go to the bioarchive, this bioarchive paper, it's there. You can see it.
I think we have the link to it from the abstract as well, didn't we?
Yeah, and it's right
It's
here.
not.
Yeah, exactly. Yeah, it's on also in the abstract. And if you want, I don't know how we are with time, but if you want, I can click on this and show you a little bit the GitHub repository. It might be
Yeah,
helpful.
that could be great if you can walk them through your GitHub repository. We have time.
Yeah.
Yeah.
Okay.
Thank you. So people can see. Yeah, we can
Can you
see the
see?
repo. Yeah.
Okay. Wait. I lost the I lost the window where I can see you. Okay. Yeah. Yeah. So I mean, that's the general idea here. There's some instructions, I guess, how to install it. It's all based uh, on Python. So okay, again, we were geared towards deep learning more. So we the main point for us was to make it super easy for people using TensorFlow or PyTorch. So you can really, with one line, load the data on that. But you can also get the data like as just sequences, right? Um, so uh, if you go to notebooks, there are examples here. I believe. Oh no, sorry. These are for. Sorry, my bad. These are for deep learning. Sorry, where did I have it? Right here. Yeah. So okay. Yeah, it's experiments, not.
Yeah. So
Yeah, so experiments, and then here we have TF, and then here is human non-data promoters. That's the data set we're working with. And so basically loading the data is just this part, or saying which data set you want is like this. And then you run this code, and then this loads the sequences. And here you can see you have train and test, how many, they're pretty much evenly, we haven't really gone into the class imbalance problem with this uh, data set, so they're pretty evenly matched. And then, okay, beyond that, we have the, um, vectorization training. This is how the training has happened. So you can see the speed here, and then this is how you evaluate it. So of course, I assume that you will probably do very different things from the beginning. So you probably will just need the first part to download the data. Uh, but it could be interesting. Okay, let's see. There's another question. Each sequence of length 251, it's exactly 251. Does that correspond to the number of features in each dataset sample? Huh. So the simple for us, at least, I assume that for you it will be different how you will do it. Oh, sorry, I will do it. I assume that for you will be different. For us, how we did it, the simplest way to embed this sequence is, since we have 4 letters, um, our first layer is essentially a one-hot encoding of length 251 and depth 4. So, you can have 0 or 1. So you have 0010, for example. And then, this 251, this gets uh, fed into this one-dimensional convolutional layers, essentially. So the the simplest way to represent the sequence is this, from our point of view, what we use, right? How many columns does the data set have? Yeah, each sample has 251 columns, and each column has a letter that could be one of 4 values. So you can represent that as a 251 by 4 one-hot encoded matrix. Um, Other people, so this is the simplest way. Other people are encoding also, using a type of embedding, like, so instead of encoding each letter, they instead use kind of words. There's no fixed word, like length or anything. But there are ways to calculate, to kind of identify overrepresented subsequences, and then measure those and represent a longer sequence as either a bag of words, or in some other way. It's not really my particular field, so I can't really tell you much more about it. But if you're interested in that, I can probably tell my student to send you, like to give me some reading materials, and I can send those to you. Yeah, but anyway, essentially the data looks like that. It's 51 characters. It's the dictionary is 4 letters. So, um, and we managed to get about um, I believe it was something like 85, 0.85 accuracy on it. If I remember
And
correctly.
I'm curious, actually, when you run your this model, which type of environment did you run it? Did you run it locally on your thing or on the cloud or something?
So I don't remember exactly where this was run, but for this type of model, so we run things on Google Cloud a lot. We have a couple of machines with GPUs that we can also use. And but also for this type of model, like this, not a huge data set. Uh, I know my students have also been running on Google Colab.
Okay.
It's Google Colab gives you a free GPU for a little, short time. So for prototyping like this, you can do it. The issue with this type of model is that you need to do if you really want to do it like properly, like and really get the best performance, you need to do hyperparameter optimization, which means that you need to train not one model, but you need to train maybe hundreds of models. And then, with bigger architectures, then it can really add up to a much longer.
Interesting.
Um,
Speaking of architecture, this is obviously out of scope of this. Do you think
right.
neural network architecture search could be very useful for this?
Yeah, I think there are some people that are working on it, but
Okay.
I like I know some people are working on it definitely. Yeah, I mean, it's partially the architecture, like for example, the number of layers. You can treat it as a hyperparameter. Like you can say, try, within a limit that makes sense. But then when you start going to more interesting architectures like multiple branches, um, combining different types of layers, it can become much more complicated. And I mean, should it should be automated at some point. I'm pretty sure it will be. Um, so yeah, that's kind of the the overall idea. I don't know if there's any more questions or
No, it was very, very informative. Thank you so much. Not just going through the historic perspective to driving us in this journey until the actual core thing. It's very useful, and you have all the resources there. I recommend everyone, before you even touch the quantum stuff, maybe run the classical stuff first to look at it. To see,
yeah,
maybe that will help you, with also benchmarking, because at the end of the day, we want to see whether the quantum stuff gives us some advantage, right? So we need to benchmark the quantum stuff with the classical stuff against
right.
classical stuff. Yeah.
And I will be very interested to see what you work on, because I'm really I mean as I said, all this, a lot of that comes from different scientific fields coming together and creating something new, right? And this kind of the opportunity here in a way, if you can I mean, I understand that now the technology is very young, but
Mhm.
maybe down the line it could be the new breakthrough, or one of the new breakthroughs.
Yeah. Yeah, that's one of the reasons why we are very excited to giving this support for like experimentations to see. Uh, um, Maybe does anyone wants to make any comment? Mahdi or Noela or the people online. Anyone wants to jump, make any comment on this? What's the first impression? Can you quantize this? I'm using the term quantize in the thing in the naive sense. Okay, some user wants to come on stage. Let's see who is this. Yeah, I'll bring you on stage. Yeah, there you go. you being invited to join the stage. Hello. Hello, Madi.
Thank you very much for the talk. It was very useful, intense information, new to me, like the genomic part, but the machine learning part was pretty clear. And I actually can see how this can directly be applied, especially that you didn't use like an extremely complex data set or a very big neural network. I think this would actually fit on a project I'm working on quantum convolutional neural networks. I guess the missing step I have in my model for that is actually going to be the second part of the hackathon, which is to embed the data into quantum states. So I will try to incorporate this in my model, but I believe like by maybe by the end of the hackathon or during the hackathon, if we could actually collaborate with people working on the data set on the encoding step, we could actually like join the two works together, and we might be able to actually make some predictions using the quantum model and see how that is compared in terms of performance with the classical model. A challenge here is going to be that like the quantum model, we have limited number of qubits. We can't make
Mm.
the model very large. I have some hope since you said the network is like 3 4 classical layers, so such network I hope could actually work on a small scale quantum network, but we'll see how that goes in training after doing some sort of initial embedding of the data. But I can see like great potential here, even if we don't get very great results from the quantum model in the beginning, but just getting results is going to be a major step.
Yeah, definitely. For us here, this challenge is so much as more about the feasibility of implementing something like this, more than whether it gets great performance. Obviously, the perform if it does hint along those lines, that's fine. But yeah, it's already a good start. And as you said, Mahdi, this thing could because it's shallow network, so it could be very suitable for a quantum setup. Yeah. Okay. Um, and I will be very the thing. I will definitely connect the teams because Marty will be working on the machine learning side on the federated stuff, but the other team will be working on the encoding. Originally, they were like separate stuff, so the people working on the federated learning they could use existing encoding already, but it will be nice to see whether whatever else the other team comes up with could be integrated with. Yeah, so I'll keep that in the mind and make an intro to the teams once we get started. Yeah, and Fanos actually is very also curious. We were talking behind on the backstage for to see how because this is I guess this Fanos is the first time you are getting this thing into quantum stuff, right?
Yeah, no, I don't have any. I mean, I've read some stuff that comes up on popular media, right? But I don't know, like, which basically means I don't know anything.
Okay.
But yeah, so I won't pretend to understand anything about it, but I know that it's something. I know it's something that will be interesting for me to learn about. So.
Yeah, we'll get a thing once the teams obviously present about what they could come up with, what they found, and yeah, hopefully you will gain some insight and most importantly interest into exploiting these things. Very often this is how someone gets interested in like in this type of emerging stuff is through like concrete like a concrete thing that's aligned with their current work. So
Yeah, I was wondering, like we already discussed that a little bit, that the benefit, I guess, of this would be much faster computation, right? Or is that not the case?
Maybe Maddie, you want to go before I jump in?
Okay, so I I'm not sure if the experiments will be of faster performance, especially at this initial stage. Um, that impact to be seen needs like to have a large scale quantum network, and then we can actually compare in that sense. But some models have shown better accuracy. Another aspect is the learning parameters. Quantum networks usually have much fewer learning parameters. When a classical model requires like thousands of parameters, a quantum model could require less than 100 parameters.
Okay.
Yeah, so that aspect actually comes in handy, especially when we do stuff like distributed and federated learning, because we actually send those parameters to a centralized server that collects data from all users. So if we are sending less parameters, that is less communication resources needed, which is
Hmm.
more efficient. So that is a pretty interesting aspect. But in terms of speed of computing or training, I think that in terms of the current networks we have, it is pretty difficult to see it clearly.
Right.
Well, one could get something like that, but it
Yeah.
is not guaranteed at all.
Sure. 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 Yeah. Of course.
Yeah, there was also some a paper. I think it was published towards the end of last year, suggesting by Patrick Coles, saying training a quantum neural network actually requires far less data, for instance,
Oh, okay.
than anything. Yeah, so that could be another. I'll share that the link to the article. Very very interesting. Far more than what people had in mind originally. Yeah.
Yeah, in that work, they say that we can get better generalization
Hmm.
while we have limited data. I'm not sure how, I mean, we do have a lot of data in genomics, but
It depends though, because sometimes there are many tasks that we have. For example, we could have the one class, as I was talking about. I touched a little bit on this class imbalance problem. For example, we have some tasks that we're working on where your positive class could be 1,000 samples.
mhm.
Your negative could be billions, but your
Oh,
positive is only 1,000. So you have
okay.
really very limited, training set.
I sorry. I think in such cases, we need at some point to start thinking about other models than just classification. So some sort of clustering of the data, or for example, maybe anomaly detection in the sense
Yes,
of detecting the positive classes for
right.
samples.
We've been trying also to work with things inspired from natural language processing, like recurrent neural networks. Um,
Do we have some
eh,
sort of series data, time series, sequence data?
well, because the genome essentially, what you have is a string of
Methods.
letters, right?
Mm.
So it's in that way essentially, you know, you have like hundreds of millions, let's say, of letters, one after another, and somehow out of all this thing, something happens, right? And life emerges. So in one of the big tasks that people, and everybody is like kind of looking at now is how to basically how to take this whole thing and annotate and say this part is this class of functional thing, this part is this class of functional thing from very few real examples that um, the problem is that because many of these functional things that I'm talking about have evolved, you know together, they could look very similar. they're not, it's not like so easy to say what is a random thing
Mm.
in a living sequence. So we have this really domain, these peculiarities of working with a real, actual kind of biological language in that sense, right? So it's really interesting in many ways. Even trying to, let's say, identify random sequences, like to produce a random sequence that is actually uh, compatible to how a genome looks, it's a difficult task by itself. I mean, if you don't just go and chop out a part, right, to actually produce it, like
Yes,
generate it.
I wanted to ask about this. Like, if we have actually limited, let's say, positive sequences in our data set, one direct way of thinking about this is, can we do some sort of data augmentation where we
Mm.
generate other similarly positive sequences and include those in our data set?
Yeah. So data augmentation is a little difficult usually in this type of data. You can do partially. You can do things like, for example, now we have these 251 letters. I mean, the 251 letters, it's kind of centered there because that's how the data set was constructed. But you could move it a little bit left, right. You could make it a little bigger, a little smaller. So in that sense, you could, in the same way that in images, they do a little bit of a rotation or zooming in out or something like that. You could do things like that. Um, yeah, but generally speaking, it's arguable how much this will help you.
Yeah, maybe because you can't make sure
You
like
can,
what you generated is not a negative one. Like,
right.
you need to distinguish.
That's the other thing. Once you reach pretty high precision, you start having the problem that your false positive. You don't know if your false positives are actually false positives or they're just things that have not been annotated, but they are part of the thing you're looking for. And this is very difficult to test for. I mean, you need to do the actual experiment then, which is expensive and difficult. So.
I see. I mean, can you please explain a little bit more on the annotation here? Like,
So imagine, like I'll give you another example from another project we had. You have, let's say, the human genome, right? 6 billion letters, and we have this 1,000 functional parts that do something. Doesn't matter. Each one of these parts is 100 letters long. Okay, and they do something. So we have 1,000, and we are trying to basically be able to scan large genomic areas. I'm talking hundreds of thousands or millions of letters, and identify like anomaly detection, right? And identify where our signal makes a peak. Okay, so we've figured out a way to do it. It's a very complicated, I mean, training scheme, and so on. Um, but now we're at a point where we have very, very high precision. So we're like, let's say, we get one false positive per, let's say, 100,000 letters that we're scanning. Okay.
mhm,
But now we're reaching a point where we're scanning actually big parts of the genome and we're getting these false positives. And when I'm looking at these false positives, they really do look like the actual thing we're trying to find, right?
mhm,
Because it has learned it so well. And now you're at the point, okay, is this a false positive? Or, I mean, I count it as a false positive for evaluating my method, but is this actually a new instance of the class that I was looking at?
mm,
Because it could very well be, right? Just
okay.
not annotated. Um, so
And
yeah, that's kind of one of the Anyway,
One
really
more
happens to
question
me. Yeah.
about the. You mentioned that you were interested a little bit in the natural language processing aspect. Our research group actually works on semantic representations and semantic extraction and semantic communications, and I am a little bit exploring quantum semantic representations also. That is related to kind of defining contextual meaning of data that we have. I have a feeling that this is a little bit relevant. This could be kind of a solution in some sense, where we actually define methods and we call it language in general, so that when we have a sequence, we can actually find its meaning, and then we can be able maybe to
Yeah,
update
yeah.
our datasets in some sense.
Yeah, I mean, that's where the thing is going, probably. It's just the problem, our big problem is that, like for natural language, it's easy, relatively easy to define these things. I mean, it's still hard, but it's easier. Like when you see a sentence, an English sentence, you can pretty much tell, okay, this is a joke, or this is a positive feeling or negative. If you see a DNA sequence, you know,
So sometimes
you have some annotation, of course, you have some things, right?
yes, you define your own language of this.
Right, right. But the problem is we don't have this definition of the language, right? It's we have some annotations, we know what some things do, but we don't have the kind of we don't have the, maybe we need to get some philosophers in here, and start defining things like that.
Yeah, this area is still kind of in a theoretical level, not
Right.
actually very implementation based. It's more on developing the theory behind this, and maybe 10 years from now, we will be
Yeah.
able to do something like that.
Yeah. Okay, I think I need to go because it's
Yep.
getting late here.
Yep.
If you, I will be on
Discord.
Discord. On
Yeah.
Discord, you have to let me know how to get.
Yeah,
You
yeah,
have to
yeah. I will
send me
send.
the
I'll follow.
address.
I'll follow up on the thing on the email. But
Okay.
thank you so much. It was
Yeah,
very
and I'll be
very.
happy to talk again. In Discord, or we can set up another chat. Like even,
Yeah, yeah,
I think
sure,
even
sure.
through there we can, right?
Yeah. Thank you. Thank you so much.
Okay.
It's been very
Thank
very
you.
insightful, and
Thank you
I
so
hope
much.
everyone else who joined, you have some interesting insights about the use cases in genomics. And yeah, I see you on the hackathon. I will